Hello friends, now we are going to see about a very interesting topic. What is immunology? Immune, this is the word came from the Latin immunus, means to be free. People survived the calamity of epidemic diseases when faced with the same disease again. So, the study of physiological mechanism that humans and other animals use to defend their bodies from invading organisms such as bacteria, virus, fungi, parasites, toxins. Coming to immunology buzzword. So, these are the very important words we hear when we see about the immunology topic. Antigen. Any molecule that binds to a immunoglobulins or T cell receptor, we call it as antigen. Means any molecule which binds to the antibody, we call it as antigen. That is very simple. That pathogen. Microorganism that can cause the disease, we call it as pathogen. Antibody. So we define the antibody as capital A and small b. This is abbreviated like this. Secreted immunoglobulin are the antibodies. Immunoglobulin, Ig. Antigen binding molecule of B cells, we call it as immunoglobulin. Vaccination. Deliberate the induction of protective immunity to a pathogen. Means through vaccination we are developing the immunity. Immunization. The ability to resist the infection is immunization. Types of immunity. We have the innate immunity and adaptive immunity. Innate immunity or inborn immunity. This type of immunity is present within the body from the birth itself. It is the immunity developed by our own body. Host defense mechanism that acts from the start of an infection but do not adapt to any particular pathogen. It is a fixed army like the thing present in our body. It will act on whatever the pathogen comes into our body. Next, adaptive immunity. Response of an antigen specific B and T lymphocytes to an antigen. Immunological memory. For example, initially we attacked with a smallpox. Smallpox antibodies are not being present in our body. So, smallpox antibodies are generated in our body with the first infection. Later, the smallpox again if gets infected, we will not be attacked with the smallpox because of this immunological memory by B and T lymphocytes. So, this we call it as adaptive immunity. This is developed specific to the pathogen or specific to the bacteria virus. Next, types of immunity, humoral immunity and cell-mediated immunity. Humoral means liquid, cell-mediated means through cells. Coming to the humoral immunity, immunity that is mediated by antibodies can be transferred to a non-immune recipient by serum. So, we can take the immunity from a serum from the other donor also. Cell-mediated immunity, immune response in which antigen-specific T cells dominate. Here, the immunity is mediated by the cells. For example, T cells. Immunological cell histology. What are the cells which comes under the immune system is polymorphonuclear means lobed nucleus. Mononuclear means non-lobed nucleus. Granulocyte, many granules seen in cytoplasm. So this is the whole picture of the blood. So we divided the blood into plasma, erythrocytes, platelets and leukocytes. For example, we have taken a sample of blood after centrifugation. So we got the three layers. So this red color region is erythrocytes, which is around 4.2 to 6.2 million per cubic millimeter. Yes, this is erythrocytes. Next, this very top thing is plasma. Inside the plasma, we have 92% of the water, 7% of the proteins by weight, and 58% of albumin, 37% of globulins, 4% of fibrinogen, 1% of regulatory proteins, and the other solutes are 1% by weight of electrolytes, nutrients, respiratory gases and waste products. This is the whole thing seen in plasma, this straw color fluid. And next, this buffy coat. So the center you will have a white color buffy coat. In that we have the platelets. Platelets will be around 12 to 3000 per cubic millimeter. And we have the leukocytes. So we 300, so total to 300,000 per cubic millimeter. In the leukocytes we have 5 to 10,000 per cubic millimeter. In that we have the neutrophils, eosinophils and basophils. These three will comes under granulocytes. Among these three also, neutrophils will occupy the major percentage which is around 60 to 70 percentage. Next is being occupied by lymphocytes, 20 to 25 percentage. Lymphocytes and monocytes are, these two will comes under the category of agranulocytes. And next, the place is occupied by monocytes, 3 to 8 percentage. Monocytes is the very bigger cell in the blood cells, very bigger cell. And we have the eosinophils, 2 to 4 percentage, basophils 0.5 to 1 percentage. This is the normal values of the overall blood. Coming to the neutral, does not stain to acid or basic compounds, which because of that we call it as neutral. Acidic, red pink, stains to acidic compounds, eosinophils. Eosinophils are nothing but acids. It stains to acidic compounds. Next, basic, blue purple in color, stains to basic compounds. So in this picture it is clear that basophils, here it is purplely stained. Eosinophils, it is pinkly stained on acidic compounds. It is violet or purplely stained in basic compounds. In neutral solution, there is no any reaction to acid or base. Cells of the immune system. Many cells of the immune system derived from bone marrow. Hematopoietic stem cell differentiation. Yes. Initially, it is pluripotent stem cells. It is being divided into lymphoid stem cells and myeloid stem cells. Lymphoid stem cells will undergo differentiation to form thymus, T lymphocytes, NK lymphocytes, B lymphocytes and plasma cells. So this is about lymphoid stem cells. And next, myeloid stem cells will divide into erythrocyte, megakaryocyte for blood clotting, and monocyte, granulocyte, macrophages. Macrophages are derived from monocytes. Components of the blood. Serum versus plasma. What is serum? A cell fluid liquid minus the clotting factors. We call it as serum. Plasma. Cell-free liquid with clotting factors of solution. We call it as plasma. When the liquid is with clotting factors, we call it as plasma. When the liquid component of the blood, absence of the clotting factors, we call it as serum. So this is again the picture of the components of the blood we have seen priorly. Lymphocytes. There are many types, important in both humoral and cell-mediated immunity. We have the B cells produces the antibodies. T cells are cytotoxic T cells, helper T cells. And we have one more cells which is also called as memory cells. Plasma cell in tissue. Fully differentiated B cells which will secrete the antibodies. Natural killer cells. Killer cells infect with certain viruses. Both innate and adaptive immunity, antigen presentation will be there. Monocytes or macrophages. Phagocytosis and killing of microorganisms, activation of T cells and initiation of immune response. Monocyte is the young macrophage in blood. There are tissue-specific macrophages also and antigen presentation will be there. Dendritic cell. Activation of T cells and innate adaptive immunity found mainly in lymphoid tissue. So, dendrite means tree-like cells. Because of that, we call it as dendritic. Functions as antigen-presenting cells. Most potent stimulator of T cell response. Mast cells. Expulsion of parasites through a release of granules. Histamine, leucotherins, chemokines, cytokines also involved in allergic responses. Neutrophils. These are a granulocyte. Cytoplasmic granules, polymorphonuclear means the nucleus contains many lobes. We can see in this picture also. The nucleus contains many lobes. Short lifespan, very important at clearing the bacterial infections and innate immunity. Eosinophils kills the antibodies coated parasites through degranulation. Involves in allergic inflammation, agranulocyte, so which is a granulocyte, and double-lobed nucleus, which is having 2 lobes in the nucleus. Orange granules contain toxic compounds. Basophils might be a blood mast cells, a cell-killing cells. Blue granules contain toxic and inflammatory compounds, important in allergic reactions. Other blood cells are megakaryocyte for platelet formation and wound repair, and erythrocyte is for oxygen transport. So this is the major lymphoid tissue. Major lymphoid tissues are, we have the primary lymphoid tissue and secondary lymphoid tissue. So primary lymphoid tissue are cell originate or mature will leads to primary, where the cells will be lymphocytes and T lymphocytes, where it will originate. So that we call it as primary lymphatic tissue. So we have adenoids. So there are different types of lymphatic tissue. There are adenoids, tonsils, we have the thymus, spleen, Peyer's patches in the intestine, bone marrow, appendix, lymphatic vessels and lymphatic nodes. So this whole thing will comes under immune system. So there is the picture of lymph node. In this picture we can able to see this outer portion is the cortex. So we have the germinal center here. So in this center we have the germinal center. This is the primary lymphoid follicle. This whole thing is the primary lymphoid follicle. And this is the afferent lymphatic vessel. This is the secondary lymphatic follicle. So primary lymphatic follicle and secondary lymphatic follicle. In the secondary lymphatic follicle, germinal center is present. We have the medullary cords and we have the medullary sinus. This is the medullary cords, this is the medullary sinus, artery, vein, and this is the efferent lymphatic vessel, and these are marginal sinuses. These are all the parts of each lymph node. So thank you very much. Please do like, share, comment and subscribe to my channel. Thank you.